Hello, and welcome to Plants in Python. My name is Bob Van Buren, and I am an assistant professor in the Department of Horticulture at Michigan State University. And in today's lesson, we're going to talk about short-read analysis using Illumina data and provide a general introduction to RNA-seq or gene expression analyses. And so the learning objectives of today's lesson are first to understand the principles of gene expression analyses in RNA-seq, so how to go from setting up an experiment down to generating the samples and doing downstream analyses. We will explain the fundamentals of Illumina sequencing and how the data is generated and how we can analyze it. You should be able to understand the distinction between mapping and quasi-mapping of Illumina data. So this is relevant for gene expression analyses, but then really any other analyses you would do with Illumina data. I'll introduce the downstream analyses of gene expression. So this would primarily include differential gene expression, gene co-expression network analyses, and also some systems biology and some modeling machine learning type approaches. And at the end, we'll introduce the frontiers of gene expression analyses in plants, which is single-cell RNA-seq. So this is an average plant cell. And so what I'm showing here is an example of a leaf plant cell. So you can see that this plant cell has tons of chlorophyll, so these are what do photosynthesis. And the average plant cell contains between 100,000 and 1 million individual messenger RNAs or mRNA molecules, among many other types of RNAs, such as long non-coding RNAs, small RNAs, etc. And a typical cell is expressing 10 to 15,000 genes at any time, depending on the cell type, the time of day, if it's stress, cell division, etc. And so these mRNAs that the plant cell is producing, it will synthesize them in the nucleus. They get translated, if we think back to the central dogma of molecular biology, they get transcribed from the DNA, turned into mRNA, and translated by ribosomes into proteins, and then these proteins function in all sorts of cellular processes. And so the gene expression profile is a really great indicator of what that cell is doing. Is it a leaf cell? Is it a root cell? Is it part of a flower? Is it stressed? Is it healthy? What time of day is it? Is it doing photosynthesis, etc.? And so mRNA turnover can be really rapid, so within a couple seconds, or it can be very slow within a couple days. And generally, expression of genes is tightly coordinated in this elegant regulatory networks that control when and where genes are expressed. So for instance, if a plant encounters an herbivore, so let's say an insect bites a plant cell, or plant leaf, within a couple of minutes, that plant will have a hormone response of, let's say, jasmonic acid, and then within maybe 5 or 10 minutes, that will initiate a downstream signaling cascade that will change what genes are expressed and how they're expressed in the cell. And so we can survey really easily what genes are being expressed, when and how, and then we can use that to answer fundamental biological questions. And so this general approach of assaying what genes are being expressed and how, we would call this gene expression analysis, or what is most commonly used now is we refer as RNA-seq, and so that stands for RNA sequencing. And so this is sort of the general workflow. This is the type of data that you encounter now, but this could change in a couple years. And so in general, you would have your samples of interest. And so in this case, this would be our biological questions. So these are tomato plants. So maybe we have a happy tomato plant and a not so happy tomato plant, and we want to see how they're different. So we would set up our experiment. We would collect some data. We would isolate RNAs from this data. So these are preferably mRNAs, but typically all kinds of mRNAs. We will select only those mRNAs that have a poly A tail. So these are going to be the mRNAs that will be translated. We will turn these into cDNA. We will do some fragmentation, some size selection, and add adapter sequences. And so these will allow us to sequence them on an Illumina machine and to provide barcodes that will tell us what the sample is, where it came from. We then take the sequence these sequence fragments, we amplify them, and we'll talk about this in a little bit, on this Illumina flow cell here, and then we sequence hundreds of millions or billions of individual reads that accumulate into tens of billions of base pairs. And then we do some bioinformatics analyses where we have our reads of interest, we remove all the sequences we don't want, and then we align those either to the genome or to gene models, and we quantify how many reads that we have, and the number of reads you have per gene will correlate to how much that's expressed. So generally a gene that's highly expressed will have a lot of reads that we randomly sequence, and a gene that has low expression will have typically low number of reads. And then we can do various downstream analyses. So this is sort of the general workflow of how RNA-seq works, and now we're going to talk about the individual steps. And so typically the most common type of technology that you would use for RNA-seq is using Illumina sequencing. And so in the previous lecture we talked about third generation sequencing that's single molecule based, and Illumina or next generation sequencing is basically a PCR type based approach. And so we start with a random library of DNA or cDNAs that we construct for each sample. And so here we have our starting molecules. This could be RNA if we're doing RNA-seq. It could be DNA if we're doing some type of DNA-seq, resequencing, maybe something with chromatin or methylation, whatever you want. It could also be a PCR amplified fragment. Then we have our DNA, and we apply 1 or 2 adapters to each end. And these adapters have sequences that allow them to bind to the Illumina machine. And they also have a unique barcode that specifies what that sequence is. So let's say we have 96 samples from different tissues, different replicates. We can give each one of those samples a unique barcode, and then when we sequence those reads, we can pull them all together, and then afterwards say which reads those samples came from. And so in general, on a typical Illumina machine, you will generate several hundred million or billions of these 350 to 300 base pair reads. And so this is what an actual Illumina flow cell looks like. And so you load your sample into here. There's hundreds of millions of little wells. You run what's essentially like a PCR reaction where you amplify each of your DNA molecules to millions of molecules. And then you add one base pair at a time. You flush the flow cell, and then you apply a camera, and each one of these clusters will correspond to a single read, and then the color dictates if it's an A, T, C, or G. And so here you can see this is really small. This is about 20 microns. And so what it would do is it would add a base pair. It would take a picture of each one of these clusters, and it would say, okay, I don't remember which colors correspond to which nucleotide, but it might say, okay, all the yellows are T's, all of the blues are G's, etc. It would record that, it would wash it, and then it would add another base pair. So this is kind of what it looks like. And so this is just showing a couple hundred, but in reality there are tens to hundreds of millions or even billions of these individual clusters here. And so this process takes between a couple hours to 48 hours, depending on how long the sequence reads are. And at the end of the day, we get millions or billions of reads, depending on the mode and the sequencing machine. And so each one of our samples from a different tissue or different plant or whatever your design is will have a unique barcode. So this is going to be a random sequence of A's, T's, C's, or G's. And then we can pull all those together. And then after sequencing, we can demultiplex our samples and say, okay, the first 15 base pairs we sequenced matches this sample that we generated, and then you can pull them into individual samples. And so in a single lane, we might get 400 million or a billion sequences. Maybe we only need 20 million per sample. So you can then use these barcodes to separate them after doing the analysis. And right now, these numbers are constantly going out of date, but you can generate about a terabyte worth of sequences per day on an Illumina HiC6000 machine. And the current cost to resequence a human-sized genome is about $100. So we're generating massive amounts of data, and we can use these if we care about the actual sequence composition itself to look for maybe variation between yourself and the human reference genome, or we can use it for RNA-seq to quantify gene expression. And so when we get these massive number of reads, they're in this format called the FASTQ format. And so what is this format? And so we're familiar with the Fasta format. So this is basically, we have a caret, a name or qualifier for whatever the sequence record is, and then the actual sequence itself. And so the FASTQ format is very similar to that, but it also includes quality information. And so here is an example of a FASTQ format. So this is what you would get off of an Illumina machine. And then there's basically 4 lines for each entry. And so the first line, that's going to start with the @ symbol. And this is data from the sequence read archive, so it's SRR. This line is going to be the unique identifier for that read. So if you remember, like reads can be, you can have billions of reads per lane. So this is going to be a really long sort of random string of letters and numbers. And then it might also say the length of the read. The second line, and this is going to be starting with nucleotides, is going to be the actual sequence of the read. That's what we care about the most. And so here you can see this string of A's, T's, and C's, A's, T's, C's, and G's. That's a length of 200 base pairs. Then the third line is the same unique identifier for that read, and then it specifies the fourth line, which is the quality score. And so when you're running Illumina data, each base pair is going to have a quality score associated with it, and that's basically reflective of how confident the machine was that that was an A, T, C, or G. And so if you remember back to the image I showed before, sometimes it's very clear that this signal is blue, indicating this letter, or it's yellow, indicating that letter, but other times it can be a little bit muddled. And so the quality scores are going to vary depending on the generation of Illumina sequencing, but each one of these letters, numbers, and random characters corresponds to a different quality score. So typically the beginning of the read will have the highest quality score, and then if it's not sure, it will typically go down as the read length continues. And the nice thing about this is when we're working with this data, we have a good approximation of how high quality that data is. And so if one read has really low quality, where maybe that cluster was near another cluster and the Illumina machine wasn't sure what that sequence is, it will tell us in this format. So this is a really useful data that we can generate here. And so these files tend to be quite large. So they might have millions or billions of sequences, but this is sort of the general format. So this is the information that we care most about. And you'll be encountering FASTQ formats throughout the rest of this course. So where is this short data stored? So when you generate this data, you would get it off a machine, you would download it either onto your local computer, or more likely you would download it onto maybe a RAID system or a high-performance computing cluster. But that data is not going to be there permanently, and so during the publication process, researchers will typically deposit the sequence data onto a public database for long-term storage. And that will ensure that if something happens to your local computer or some type of external hard drive, or maybe your high-performance computing cluster, that data is not lost forever. And so there are a number of different places where you can deposit this data, but where you'll find the vast majority of it is what's called the Sequence Read Archive, or the SRA, on NCBI. And so we're familiar with NCBI from the previous lessons. And so the NCBI currently has petabases of DNA, and so a petabase is this many base pairs. So it's just massive amount of sequences. So this ranges from whole genome sequencing data, various RNAseq or gene expression data sets, DNA-seq, etc. Just tons and tons of data. For example, for the Arabidopsis, the model plant, there's probably on the order of 40 to 50,000 individual RNA-seq experiments, and it's all held on this public database. And so in this lesson, we'll actually be downloading some data from the Sequence Read Archive and doing some quality assessments and some downstream analyses. But this is sort of what it looks like. And so if you type if you go to the NCBI website and you go to the Sequence Read Archive, you can type in your species of interest or a stress or something. In this case, this is data from Iragrosis teff, or this Ethiopian grain crop teff. And this is data from teff leaves, and it's a waterlogged sample. So this is basically a flooding sample. And it will tell you, it will give you this unique identifier that specifies what that data is, the Illumina machine that was used to run it. So this is kind of an older machine, the Illumina HiSeq 2000, how many spots or how many individual reads it generated, the total number of bases, the design, the institution that submitted it, the species, and then this is the actual number that we care about. We need to use this to actually download that data, and then again it will tell us how many individual reads there are. And so you can search on here for your favorite organism, for your favorite stress or tissue or condition or whatever you want, and there are probably on the order of hundreds of thousands of samples for plants. And so there's this massive treasure trove of data, and typically in a single paper, researchers only skim the surface. So this is a great place to go to find data and to answer whatever question you're interested in. Okay, so when we get this raw data off of an Illumina machine, typically it will contain some low quality base pairs. And so these can be, as the machine is running, it takes between 10 and 48 hours for each Illumina run, and as it goes, the quality goes down with time. And so you are adding a single base pair, you're washing that column, you're adding another base pair and washing it, you're heating it, you're cooling it, and so with time, the DNA molecules can degrade and the quality goes down. And so we want to remove base pairs that are low quality, where we don't know for sure if it's the letter that the FASTQ file says it is, and so we need to get rid of those. And you can also have leftover adapter or barcode sequences, and we want to remove all those because those won't be found in our genome, and low quality bases that are incorrect, they affect the downstream analyses. So reads need to be what we refer to as trimmed before processing. And so here's an example of, this is a library of Illumina data, and as you can see, this is the quality score, so higher, 40 is about the highest, and then it goes down to 0, and so generally the higher the better, and we would say anything above around the 30 mark is going to be considered high quality, and you can see as we go across the individual base pairs, the quality tends up going down. And so we might want to trim reads to about this length, or trim all the reads that are below this threshold here, because as the quality score goes down, the confidence that that read has that nucleotide also goes down. So we need to do some processing before we move forward with the analyses. So there are a number of, like anything in bioinformatics, there are a number of leading programs that can be used to trim reads to remove either low confidence sequences that's based on their quality score in the FASTQ format that we talked about, or residual adapters. And so these can either be applied, so we either say this is the barcode that we use to make this library, or some programs will automatically search either their database or they'll search certain sequences or k-mers and identify them as putatively adapters. And so there are a couple programs that are most common for this. CutAdapt is probably one of the older ones, and then I think the two that are most common right now are Trimmomatic and FastP. All these programs are a little bit different, but they'll generally give you more or less the same result. In our course, we'll be using this program FastP because it is an order of magnitude quicker than the other programs, but in general, they'll all give you about the same amount of data. And how much you trim depends on the quality and how old your data is. So typically, older data, you're going to need to trim a little bit more than newer data where you'll have to trim less. So this is kind of the first step of we get our data, and then we need to get rid of all the low quality data. So what happens after that? So we've generated our sequences, we have our experiments, we've trimmed the data. Now what do we do? And this is sort of the central point of RNA-seq analysis is what we want to do is we have our genes or genome here. And so here's an example of two transcripts. This is human genes here. So basically each one of these is going to be an exon, and then we have some introns here. What we want to do is align our Illumina reads against the genome. And so we can do this in one of two ways, which we'll talk about in the next slide of alignment or mapping versus quasi-alignment or quasi-mapping. And so the fundamental sort of theory behind RNA-seq is that the number of reads corresponds to how much that gene is being expressed. And so when we make our RNA-seq library, we're sequencing a random pool of RNA molecules. And so a gene that has high expression will generally have a lot more RNA-seq reads mapping against it than a gene that has low expression. So I don't know if you can see here, but this gene is 300 and this would be 800. And so we can use that as a proxy for how much a gene is being expressed. And so we can't really compare the number of reads between any two genes or any two samples because genes of different length, so longer genes will tend to have more reads that align to them than shorter genes just because of random chance. And individual sequence libraries, you might have 10 million reads in one sample and 20 million reads in another. So we need to normalize these to get so that we can actually compare between experiments and between genes. And then we'll normalize this count and then use this count as sort of a raw input for downstream analyses. So this is kind of the fundamentals of how this works, where we have our gene or genome, we align the reads. We don't care about the sequences for our context, we just care about how many reads are aligning like tick marks to each gene, and then we take those counts and use those as downstream inputs. And so there are two main ways that algorithms work to align or to count basically the number of reads that are aligning to a gene or a genome. And so there are many different ways to analyze gene expression data, starting from how the reads are aligned / mapped against the genome, how we identify genes that are differentially expressed, and the types of downstream analyses that we do. And just like anything in bioinformatics, there's lots of software, there's differences of opinions, there's different statistical thresholds. But fundamentally, there are two different ways that we can do this. And so the first way is through literal mapping. And so mapping-based programs will take our reads that we've generated and find the exact position that they map to in a genome or a gene model. And so these programs will identify the region in the genome that they map to, run a local realignment, and then record the exact coordinates and any differences between the genome and a read. So this produces a very large output file called a .sam file. And so this has the read itself, the sequence, and the sequence against the genome, and any differences between the read and the sequence. So this is very accurate, but generally very slow. So we're taking every single read, we're trying to figure out where in the genome it aligns to, and then recording the exact positions. This type of mapping is essential for DNA-based analyses. So if you're trying to look for variation in sequence between a reference and an unknown, or maybe you're doing some type of chromatin-based data, or maybe some methylation data, it's really essential there. But for gene expression, we don't care about the sequence context at all. We only care about basically how many reads are aligning. And so if you want, you can do a mapping-based approach, but we can also take a bit of a shortcut and do what's referred to as quasi-mapping or pseudo-alignment. And so instead of calculating the optimal alignment and the exact overlap between a read and a genome, a pseudo-alignment or quasi-mapping based program will use a hash table to figure out which transcript the read is from without doing the computationally intensive alignment. And so what they'll do is very similar to the first step here. It will create this hash table of just the gene models and see what the best compute what the best alignment is and which gene model that RNA-seq read is mapping to best. And then instead of doing the second step where it will run a local alignment and output the exact matches, it just basically makes a tick mark and say, this RNA-seq read maps to gene model X, and then it's done with it. And so this is not quite as accurate. If you have a polyploid or a genome with a lot of gene duplicates, it's not quite as good as mapping, but generally it's almost as good, and it's several orders of magnitude less computationally intense. And so pseudo-aligners output a table with counts for each gene instead of this massive SAM file, and these programs run ultra fast. So they can process millions of reads within a few minutes. So we'll actually be doing this in this course. And they probably run about 10 times faster than a mapping-based program, and it produces a lot less intermediate files. And so you can use either of these approaches for RNA-seq. They'll both generate pretty good results. Mapping might generate slightly better, but if you are working with massive amounts of data, quasi-mapping is probably the way to go. Okay, and then in terms of programs, there are a lot of great programs for doing mapping, and we'll actually need these if we're going to do some type of analysis where we care about the sequence and where it's aligning to, such as variant discovery. And so I would say the three most common programs are probably Bowtie2, BWA, and HiStat2. So these are programs that are optimized to be as efficient and as quick as possible. And if you're interested, you can read more about these. And then the most common programs for quasi-mapping or pseudo-aligning are probably Callisto and Salmon. And we'll actually be using Salmon in the activity-based in this lecture. So again, there are a lot of different programs. These programs will probably change with time. If you watch this video 5 years from now, these might be out of date. But there's constantly new programs that are better, more efficient, faster, more accurate that are coming out. But as of right now, these are probably the best programs to run. Okay, so what do we get from this? And so after mapping and quantification or pseudo-alignment, we're left with a read count table consisting of the number of RNA-seq reads mapping to each gene model. So this can be a raw count where it's basically saying 50 reads aligned to this gene, 35 aligned to that one, or it can be a normalized number. And we'll talk about that in a little bit. But basically we get this output. So this is example from a human genome, which is why all the genes have these curated names. And so each row is going to be a single gene. So each one of these is a different gene in the human genome. And each column is going to be an individual sample. So this would be KD2, KD3, whatever. These could be whatever you want. This could be maybe leaf replicate 1, leaf replicate 2, leaf replicate 3, and then root 1, 2, 3, or it could be healthy leaf and sick leaf, whatever your experimental design is, and then it'll be their gene expression values for each of these. And typically with gene expression, we want to have a number of replicates, and that allows us to better quantify the variation and to do differential expression and other downstream analyses. So we can have raw counts, which are just the number of reads that are aligning, and that's useful for some programs and some calculations, or we can normalize this data by taking into account the length of genes. So longer genes, as I said, will have more reads that align to them, because if we're randomly chopping up RNAs, a longer gene is just going to be more represented in the pool. And then we can also normalize by the number of reads we have in each pool. And so you try to get the same number of reads when you sequence these, but it's very common to have like 20 or 30% variation. So sample 1 might have 20 million reads and sample 2 might have 25 million. So we're going to normalize by both these factors and produce this normalized value that's RPKM, or reads per kilobase of transcript. So that's normalize small genes versus large genes per million mapped reads, and that's to normalize differences between libraries. And so raw counts don't take any of this into account, and then these normalized values, such as RPKM, can be used so that we can compare genes to each other and then genes across samples. And so this data frame, this is what we're going to be working primarily, this is what you care about. This can be used for downstream analyses, such as differential gene expression, gene co-expression analyses, or various modeling, machine learning, or systems type analyses. Okay, so let's first talk about differential gene expression. So this is kind of the fundamental and sort of the simplest analysis that we can do. And so again, maybe we have our happy plant and our not so happy plant. So in this case, this is a happy, healthy tomato plant and a drought-stressed tomato plant. So maybe we're interested to know what genes are controlling drought stress responses. This could be your favorite pathogen. It could be a knockout where you have maybe a gene that's knocked out and you want to know what things it's interacting with. It could be any design you want. And so we would collect RNA from leaf tissue of both of these and then run it through the pipeline and then do differential expression. And so we want to develop a statistical framework for classifying genes as differentially expressed between any two conditions. So we need to figure out what the threshold is of saying a gene in happy plants is differentially expressed from a gene in stressed plants. And so there are a lot of programs that can do this. There are a lot of different statistical models that we can do. We won't get into that today really, but the program that's probably the most common is DESeq2. So this program is based in R, but there are a number of programs that we can use. And then we essentially get this plot of differentially expressed genes. And so here they're plotted by their log fold change, where these genes would have are lower expressed in one sample, and maybe these would have less expression in happy plants, and then these genes would have more expression in the happy plants controlled to stress, and then genes down here that have, this is the log p-value, so these would be not significantly differentially expressed, and then these genes that are in blue or in red would be considered differentially expressed. And so we apply the statistical model to identify genes that are significant, and generally genes that are significant are going to have either low variation between replicates and a small fold change, or a massive fold change and have higher statistical significance. And so from this, we could say maybe there are 5,000 genes that are differentially expressed between this happy plant and this not so happy plant, and we can do various downstream analyses. So maybe we could look at what sort of genes are involved in this. So we could run a gene ontology or KEGG pathway enrichment, where we look at the types of functions that genes that are differentially expressed are involved in, or the types of biochemical pathways that they're involved in. Or maybe we could do functional characterization, where we say I'm interested in the top 10 genes that are different between a control and a stress plant, and then try to see what those are involved in. Maybe we could go back to comparative genomics and see what the genes are in maybe the model Arabidopsis plant, et cetera, et cetera. So this is really nice, but the downside of this is that if we only have two comparisons, if you're looking at an extreme phenotype such as drought stress, you might have 5,000 genes that are different between these two plants. And so where do you go from there? You can't do single gene characterization of 5,000 genes. So we can do more elegant and sort of fancier approaches where we're collecting a lot more time points, and we can look at how genes are expressed with each other, and then use that to generate a more robust list of differentially expressed genes. And so if we have lots of samples, we can run what's referred to as a gene co-expression network approach. And the goal here is to see which genes tend to have similar expression patterns between all of our samples. And genes that have similar expression patterns are more likely to be under the same transcriptional control. And so if we're interested in transcription factors, or these are regulators of gene expression, we might want to run a co-expression analysis and see which genes tend to have similar expression profiles, and those could be involved in processes that I care about. And so the general way that a co-expression network works is we have, again, our expression matrix, where we have our samples here as rows and individual genes, or sorry, our samples here as columns and individual genes as rows. Typically, the more samples you have, the more power you have. If you just have 3 samples, you're not going to have a lot of statistical power because you'll have a lot of spurious data points. But if you have 10 or 20 or 100 different samples, you get a much better survey of how genes are interacting with each other across all of these samples. We can run a Pearson correlation, where we basically calculate between any 2 genes how likely those genes are to have correlated expression patterns. And so if we compare a gene against itself, it's going to have a correlation of 1 because they'll have the same values. And then we can compare it to any one gene. So here's gene 1 against gene 2. And so you can see this correlation is very low. So those genes tend to not have high correlation, whereas maybe gene 6 and gene 2 would have a 0.99. So these genes are highly correlated with each other. And then we get this similarity co-expression scores. And then we can take this, we can sort it by a threshold, and then we can build this network adjacency matrix where we convert these numbers to zeros and ones, where any number that is above our threshold, so maybe our threshold is 0.9, or in this case it's 0.8, we would give that a 1, and then we can cluster these based on this adjacency network. And so here we would say that these genes 5, 6, 10, and 2 tend to be very highly correlated with each other. So if we go back to this matrix here, so we see gene 5, the Pearson correlation with gene 2 is very high. So we have 5 is connected to 2, and so forth. And so this can show us which genes tend to be co-expressed with each other. So genes that have a high Pearson correlation are going to be expressed at the same time in the same tissues, and they're likely to be under transcriptional control. So we would say that these are sort of 2 independent network modules, and these genes could have different functions. This is an example with 10 genes, but of course a genome is going to have 10, 20, 50, 100,000 genes, so it's really a lot more complicated than this. And so what we end up getting, this is sort of an example from a human gene co-expression network, is we calculate these Pearson correlation statistics against all of our samples, and then we can cluster them based on their function. So these genes tend to be highly co-expressed, and maybe these are involved in ribosomes or ribosome translation. We have some things involved in metal ion homeostasis, the cytokinin or cytoskeletons, immune responses, mitochondria, et cetera, et cetera. And so what we could do here is run this co-expression analysis, and then maybe we have these genes, if we're interested in immune responses, we could then see how these genes are associated with each other in some downstream analysis. And so again, this is kind of another representation that's a little bit more defined. And so we run a Pearson correlation to see like how individual genes, how their expression compares to each other. And so like in this case, gene 3 and gene 6 have really high R-squared, so they tend to have highly correlated expression. We can then we can construct this network here and then see which genes tend to have the highest correlation. And so again, gene 3 and gene 6 have this sort of linear relationship where if gene 3 is expressed highly in one sample, gene 6 is also highly expressed in that sample. So then we can create what are referred to as modules based on some hierarchical clustering or another algorithm. So we might say that these genes tend to have similar co-expression, so they would be grouped together. We could identify this as a module of all these genes being expressed together. And then we can do some regulatory network identification. So we look for the transcription factors or the genes that control when and where things are expressed. Those might be of interest to us. We can do some differential co-expression analyses. So maybe we build a network between healthy and stressed, or between maybe leaves and roots, whatever we want. We can do some sort of enrichment analysis where we might identify a hub and then say what's the function of this hub. So we would run maybe gene ontology analyses or KEGG pathways and see what the biochemistry is. We can identify hub genes. These are genes that are likely to control other genes. Or we can do some sort of guilt by association where, let's say we identify this hub here. We maybe know that this is involved in some disease and we know the function of these 4 genes, but maybe we don't know the function of this gene, and we can then do some single gene characterization here. So kind of just providing an overview of co-expression networks, but there are a lot of different algorithms, there are a lot of parameters, and in general the goal here is to maybe gene expression or differential expression might give you thousands or tens of thousands of differentially expressed genes. But if we layer on information in a co-expression network, we can get down to these much smaller interactions that could be of significance. So this is a way of narrowing down our genes of interest. Okay, so those are kind of some examples of where we are in the current sort of expression analyses. You can also do higher order prediction, machine learning, modeling, that sort of thing. You can incorporate gene expression data with other types of data sets, such as metabolites or proteins. You can correlate it with maybe various phenotypes or chromatin data, methylation data, etc. We won't get into that today, but this is kind of where the field is moving, and the field is moving into single-cell RNA-seq. So what we had talked about before was sort of bulk RNA-seq, where maybe we would isolate a bunch of RNA from a leaf sample of a happy tomato plant and a stressed tomato plant, and then look at what's different. But in general, individual cell types will have different responses. So if we think about a leaf has cells that are doing photosynthesis. They have cells that form in structure. Some of them form the epidermis or the outside layer of the plant. Some of them would function in maybe the vacuole or the phloem. Sorry, not the vacuole, the phloem or xylem. They might function in transport, etc. So each individual cell type might have its own separate response. So maybe we want to see what individual cells, how they're responding and how they're different. And so this is what we refer to as single-cell RNA-seq. So this technique was pioneered in human systems and in mammals like mice, and now it's becoming adopted into plants. And so the sort of general workflow, this is data from a plant paper, is maybe we would have our mock and maybe we're looking at drug addiction in mice, so that these mice are getting morphine. We would euthanize them, dissect this little part of the mouse brain here, and then we would take all that clump of tissue, dissolve it into individual cells, where this is looking at a hemocytometer, so each one of these is going to be an individual cell. And then we would perform what's referred to as DropSeq, where we take each one of these cells, we run it through in one channel, we run a set of barcoded beads into another channel, and then we have a suspension of lipids in another one. And what we're basically doing is we're creating these tiny droplets where ideally we want a droplet that has a single cell from our organism and a single barcoded bead, and then we create, and then you'll have a bunch of ones that don't have either of them, and then we basically make a tiny RNA-seq library in each one of these individual droplets here, and then we sequence those, and because each droplet has its own adapter or its own barcode, we can then identify them as individual cells. And so here what you would do is you would isolate your sample, you would run it through this DropSeq, and I'll show a video of this a little bit later. You would again do this high throughput Illumina sequencing, you would align it, and then what you end up getting is each one of these dots here corresponds to a single cell. And so we'll talk about this a little bit later, but this dimensionality reduction, where we're basically looking at the expression profile of all the genes within an individual cell, and then we can use that to cluster them into cells that have similar expression profiles. And so this type of dimensionality reduction is called t-SNE, which we'll learn about a little bit in the next video. And basically, each one of these dots corresponds to a single cell, and we can cluster them based on their function. And so maybe this big group of cells would be neurons in the rat brain. I don't even know what some of these are here, but maybe you have some endothelial cells, macrophages, microglia, whatever. These are just different regions of the brain or different cell types in a brain. And then you could look to see maybe how these individual cells correspond in maybe a mock treatment versus morphine or drug treatment. And so this is really powerful because instead of saying, how is this bulk tissue responding, we can say, how are individual tissues responding in a tissue-specific manner. And so here is a video from YouTube. This is actual DropSeq in action here. And so this is a microfluidics device. And then we have our lipid suspension going in here. And then here are each one of our cells that are kind of rolling down here. And you can see these individual cells being captured by these droplets here. And so you do these things with a certain dilution, so maybe only 1 or 10% of our cells actually have the droplet that we want, or that have a bead in them and an individual cell, and you can go down and make an RNA-seq library from each one of these. So this is kind of the emerging frontier of where this is going. Okay, and so this technique is being applied pretty rapidly to plants as well. The major challenge with plants is that it's a lot more difficult to isolate single cells from plants than it is from animals, and that's because plants have a really robust cell wall. So it takes a lot of enzymes, a lot of effort to dissolve plant cells from each other, but there have been some really exciting papers recently, like this one from 2019, where they were looking at single cell RNA-seq in Arabidopsis roots. And so here we're showing these are the individual cell types. So each one of these corresponds to a different cell type in an Arabidopsis root. And then this is a culmination of about 12,000 individual Arabidopsis cells. Each one of these points here is a single Arabidopsis cell. And then we've used this dimensionality reduction where we're using the expression profile of all the genes in Arabidopsis to group these cells. And you can see that individual cell types tend to have similar expression profiles. So maybe this orange one would correspond to an epidermis, and here's where those orange ones are. Maybe this would be like a root hair cell, and those could be down here, et cetera, et cetera. And so maybe you care about these cells that are dividing here, and you could look for these in this clustering. And so you can use this to identify potentially new cell types. And so this has been used in humans to identify new cell types in the brain. It can be used here to potentially identify new cell types in plants that we don't know about based on physiology or development alone. You can use this to see how cell types are responding in a tissue-specific manner. So maybe if we have a pathogen in the soil here, how the epidermis is perceiving that pathogen is going to be very different than some of these internal cell types or these transportation cell types. So we might care about that. You can do a comparison of a control versus a stress, and we can really get down at the single cell level of how genes are being regulated and how tissues are responding. So this is a really exciting time to be in plant sciences, and increasingly we'll look at the single cell responses. So maybe in your career or your graduate studies, you'll be working in some of the single cell data. So this is kind of where the field is going. Okay, so just to summarize what we talked about today, again, we talked about lots of different things, but I just want to provide a general overview. But you could spend weeks talking about this, maybe even a whole course talking about gene expression analysis and some of the downstream processes. But hopefully today you gain some foundational understanding of these things. And so learning objectives, what we should come out from this lecture is an understanding of the general principles of gene expression analysis in RNAseq, the fundamentals of Illumina sequencing, how the data is generated, how it's analyzed, understand the distinction between mapping and quasi-mapping or pseudo-alignment of Illumina data, the benefits, how each of these can be used in downstream analyses. Wanted to provide a brief introduction of the types of downstream analyses that we can do. So here we've highlighted primarily differential expression and gene co-expression network analyses. Again, I just provided kind of skim the surface here, but you could spend weeks learning about these, and there are other learning resources that you can use. And then finally, just have some idea of where the field is going, and that's in this area of single cell RNA-seq analyses. And so hopefully you learned a lot in this lecture, and then the remainder of this notebook, what we will be doing is actually processing some RNA-seq data. So we'll download data from the sequence root archive, process it, quantify gene expression, and then we will be learning about dimensionality reduction and how we can use that to assess the quality of our RNA. And then the preceding notebooks, we will be doing some downstream gene expression analyses.